Hello.
Hello, hi. How are you?
Good, how are you?
Good, yes. Just wait for a few more people to join.
Sure.
We're also on Facebook, so hopefully there should be some people
Nice.
as well.
Should I try to maybe share my slides?
Yes, that'd be good. I have to ask. There we go.
Oh, yeah.
That works. Okay. Give you a short introduction. Yeah. Okay.
All right.
Okay, hello everyone. My name is Ben. I'm the Secretary of Biosop. I'm giving a short introduction for our last speaker of Michaelmas term, Shrike Zhang. Dr. Zhang is an assistant professor at Harvard Medical School, where he runs the Engineering Living Systems Lab. He'll be talking today about his lab's research into novel methods of tissue fabrication using 3D printing. Dr. Zhang will also discuss how his recent work relates to the studies of tissue regeneration and human microtissue models to promote personalized medicine. Okay, so I hope everybody enjoys the talk. And if you have any questions, then please wait until the end for the Q&A or post your questions in the chat and we can come back to them later. So over to you, Dr. Zhang.
All right. All right, thank you for the invitation. So yeah, again, as introduced, I'm going to talk primarily today about our work in the last few years about 3D printing put towards the fabrication of human tissue and tissue models. So a little background to start with. So we're interested in the drug development process because, as you know, this is a long and expensive process. So for example, in the US alone, right? So in order to bring a drug formulation from tens to hundreds of candidates to the market, you have to go through 10 to 15 years of the process and also multi-billion dollars for that to happen. So that's a very long and expensive process. So the issue with that is because conventionally people use different models for drug testing. For example, people use these two-dimensional and static monolayer cell cultures for drug testing. And we all know that in the human system, there are essentially no tissue or organ that is flat and is static, right? So meaning that a lot of the drugs that are tested positive on this conventional models don't really translate into human system. So that's one of the issues with these models. And people also use animal models, for example. They are usually pathologically or physiologically relevant, but then they are also having issues. For example, they still possess genetic difference from humans, so that a lot of drugs that are tested positive in these animals, they also don't translate. And also they have issues of, for example, lower throughput and higher costs. So those are also limiting their translational potentials. So that's why in the past 10, 15 years, people are really trying to develop these high content, higher throughput models of human tissues and organs in vitro for us to be able to screen drugs or personalized medicine in a better way. So these are basically so-called organ or chip systems where each of these organs are made onto these little microfluidic devices. And so now you not only have the three-dimensionality of the organs, but also you have the dynamics built into these devices as well. And more interestingly, you can actually link these different tissue or organ models together in the way that these different organs are connected in the human system. So now you can start to also look at their interactions and also interactions with different drug molecules and all the pharmaceutical agents you put into the system. All right, so that's basically near-term goal of using these tissues for drug screening applications. But of course, as you can imagine, these different biofabrication methods can be all applied to the conventional sense, tissue engineering and the regenerative medicine kind of scenarios. So basically, the rationale for this is there are many people that are on the tissue and the organ donor list, right? But then the actual amount of people who receive the actual donor organs are very small comparing to those on the waiting list. So one way that people were thinking about is how can you actually engineer these human tissues and organs so you can implant them back into the human body for generation instead of having to rely on donor tissues and organs. So then, as I mentioned, you can imagine these different tissues that you can fabricate can also be eventually applied for implantation purpose. But of course, this is longer term goal because you have to make sure that everything is perfectly aligned with the human body so that you don't elicit adverse effects of this engineered human tissues and organs that you engineer. Right. So then those are basically also what we are interested here in the lab. And in order for that to happen, we are actually focused on a bunch of different technologies, including, for example, biomaterials, basically how you can engineer these materials for creating these tissues in a better way. And of course, biofabrication, including bioprinting that I'm going to talk about today, to position this different tissue, different cells and biomaterials in a way that you can recreate the tissues and organs. And further down the road, we have been developing these biomedical devices that can allow better maturation and culture of the engineered tissues and organs. And finally, we have also been interested in bioanalysis. Basically, once you have these tissues and organs made, you want to actually know how they behave, or if you, for example, infuse drug molecules into the organ chip devices, you want to also know how they interact, right? So that's why we have been also interested in these bioanalysis ways to allow us to do real-time analysis of these interactions. So one prominent example is we have been really trying to combine, for example, bioprinting with these organ chip devices so it can basically recreate not only the three-dimensionality brought by the bioprinting method, but also the dynamic flows or other mechanical kind of cues in these engineered tissue models to really allow you to create high content, higher throughput models of human tissues and organs for drug screening and other purposes. So today I'm going to primarily talk about bioprinting as a part of biofabrication. But before I go to that, I want to also briefly mention the other two aspects, bioreactors and also biosensing. So for example, we can engineer these different bioreactor or microfluidic device systems where you can host these engineered human tissues and organs in these little chambers, and you can start to perfuse them in a way that you can improve their functionality. For example, this is a simple structure of the bioreactor, and you can actually create multiple chambers hosting multiple tissues in the bioreactor. So you can now start to also look at their interactions, for example, metastasis of tumors within this device. And you can also create this mechanical kind of actuations in the device for modeling these biomechanically active tissues in the human system, like vasculature or other tissues. And of course, you can put them together in the way that they are linked in the human system, so you can start to look at their interactions. And for bioanalysis, as I mentioned, that's also important because you want to actually know how these tissues are behaving or how they're interacting with different drug molecules. So we have been also developing these different types of the biosensors, of course, in collaboration with different groups to allow us to provide in situ, continuous and non-invasive analysis of biomarkers, for example, that are secreted by the cells or electrophysiological behaviors of the cardiac tissues or neuronal tissues, and also a bunch of imaging kind of techniques to allow higher content imaging capacity for this engineered tissue and organ models or their other versions. All right, so then coming to the focus of today's talk, bioprinting. So again, as we know, the human tissues and organs are complex, right? So these are some exemplary human organs: heart, brain, gastrointestinal system, and all the other organs. And below are actually their corresponding microscale tissue building units. And we can see from these images that these are very, very complex, having multiple cell types coming together, having multiple ECM, extracellular matrix coming together in the right architecture in order for the macroscale tissue to be functional, right? So the challenges here really are to how to mimic the complexity of the native tissue microarchitecture and how to reproduce the synergistic interactions of the multiple cell types. So with that in mind, there have been many years of efforts on making these tissues with different ways. For example, people use these scaffolds, basically like matrices for allowing tissue or cell spatial arrangement and tissue growth. And people can, for example, knit these fibers into three-dimensionality for modeling the tissues. And there are some ways for like self-assembly based approaches to allow larger scale tissue fabrication. So these are, as you can imagine, these are more manual approach or semi-manual approach, meaning that if you want to make one scaffold, that's okay. If you want to make 10, maybe okay. But if you want to make 1,000 scaffolds, that's going to become a major issue because first, it's labor intensive since it's all manually made. And second, the more significant issue is that if you manually make all these different scaffolds or tissues, then if you make thousands of them, they're all going to have different properties, meaning that the reproducibility is actually not sufficient, right? So that's why in the past maybe 10 years, people are really trying to develop this 3D bioprinting approach. And this is actually very like one of the simplest ways of bioprinting called extrusion bioprinting. So as you can appreciate from the video that it's a fully automation process, right? So basically you have a nozzle in there that's extruding the bio inks or biomaterials and cells. And then by controlling how the bio ink is deposited and also the how the movement of the nozzle or the substrate is, you can basically position these cells and biomaterials in 3 dimensions without any human interference, right? So that's a very automation process and potentially allows a larger scale fabrication and also reproducibility for the different tissues that you fabricate. So then we have been really trying to develop essentially every aspect of bioprinting, all the way from hardware, software engineering to biomaterial or bio-ink designs, and all the way to downstream applications. So here's some synopsis of the different areas that we work on within this bioprinting field. So for example, we have been really interested in multi-material bioprinting, as mentioned, tissues are complex. So basically you want to design these multi-material bioprinting methods to allow us to be able to put different cells and biomaterials together in the way that they arrange in the human system so you can recapitulate their functions and structures. So we have also been interested in a bunch of different ways to engineer cannular tissues or tubular tissues that are also widely present in the human system, right? So like vessels and other tubes. And as I mentioned, we are also interested in bio-ink and bio-ink processing methods because you have to basically allow the bioprinting to start with using bio-inks. And then after printing, you have to also make sure that the embedded cells or tissues are functional and viable. So this is actually a very important component as well. And then the last part is minimum invasive bioprinting or in situ bioprinting, basically like how to directly print on one areas to allow in situ regeneration or healing of the tissue. That's also one thing that we're interested in. So I'm going to briefly go through the different aspects of things that we have been developing in the past few years without going too much into details, but more exemplifying the different capacity that we have with these different bioprinting methods. All right, so one example, or the first example I'm going to give is this so-called rapid multi-material bioprinting. So in this method, we actually designed a nozzle where you can deliver the multiple bio-inks from multiple channels, but a single outlet. So then by controlling how these bio-inks are delivered and how the nozzle or the substrate is moving around, you can actually very nicely tune the properties of the materials that you print, right? For example, you can start with a single material, and then each turn you're adding another material, and eventually you can simultaneously print all 7 different types of materials in a single printing process, right? So that's giving you a lot of controls over the biomaterial deposition in this unique setup. So for example, you can then print a little more complex structure hardware batch with all the different materials in there. And one of the advantages here with this bioprinting method is, as you can imagine, so we call it rapid multi-material bioprinting because we have multiple channels coming together into a single nozzle, meaning that if you want to change a bio-ink to print, you don't have to really change the nozzle, right? So you have to basically just switch your channel of extrusion so you can now start to print multi-material constructs. But in a conventional setup, which is based on multiple nozzle platforms, right? You have to physically change your nozzle when you change to a different bio-ink, meaning that the time for changing the nozzle can actually be as much as the printing process itself, which can be a lot of waste of time. So this is actually something that we developed to allow a very fast multi-material bioprinting without having to exchange nozzles. So then you can start to print more complex structures, for example, multi-material bioprinting of cubes or rings or slabs of different materials, and you can print a whole like human-like structure with as many different types of materials as you want. Right? And as you can see from the videos on the right, these printing processes were pretty smooth and continuous without having to change in nozzle, like when you deposit different biomaterials or bioinks, right? So of course you can create the hierarchical structures as well in the V directions. And then those are acellular structures. And by tuning the properties of bioink, you can also now start to incorporate cells within these bioprinted structures, right? So here, for example, is the heart-like structure and the kidney-like structure that they printed with different cell types. And you can appreciate that each color here actually represents a different cell type, right? So they're labeled in different colors. And by looking at this graph, you can also see that if you properly tune the bioink property, you can actually maintain viability of the cells in a pretty high state. They're all 70–80% of viability after 7 days of culture, which is pretty good, and they will also proliferating over time. And now you can start to print more complex gradient structures as well. So to be very brief, one example of application is in collaboration with some colleagues in Netherlands, right? So Jay here, we were able to print a little, we call mini brain model, where you can then model these brain tumors, in particular glioblastoma, and then start to use that for drug screening for this brain tumor kind of disease in vitro, right? So what happens here is that you can actually print a little brain structure that's about a few millimeters in size, and then into this brain tumor structure, this brain structure, you can actually print a little volume of the brain tumor, right? And the brain, little mini brain that you printed initially were also containing these immune cells. And now you can start to look at how the immune cells are interacting with the surrounding, with the little volume of the brain tumor cells in 3 dimensions. And we did show that if you compare 2-dimensional monolayer culture of cells and 3-dimensional bioprinted models, they're actually very different. With 3D printed, bioprinted volumetric models, much more biomimetic than the 2-dimensional models in the sense of cellular responses, right? And then you can start to look at all the interactions. Again, don't look at all the details, but the take-home message here is that you can then start to have different configurations of the bioprinted little mini brain and the mini glioblastoma models, and then start to look at all the interactions and the interaction with drug as well, right? So then you can start to look at, for example, if you infuse the incubate these little mini brain tumor models with different drugs, you can also start to look at how the drugs are killing the brain tumor cells in a dose and time dependent manner, and you can then register those to the human responses as well at the end, right? So these are basically higher throughput, high content models of human brain tumors you can print for improving drug testing efficiency. All right, so those are based on a method called extrusion bioprinting that I mentioned. So those are one of the simplest ways of bioprinting. So there's another way of bioprinting called digital light processing. So that explanation here. So what happens here is that instead of having a nozzle that's extruding the bioink, here you have a little reservoir, like a tank. And in this tank, you have this bioink that is photoactivatable, meaning that if you shine a pad of light at the bottom of this tank, it's going to cross-link or stabilize a layer of the bioink into a specific structure that you want. And then you can move the stage up, right? So then that's going to expose the bottom of the tank to another layer of the uncross-linked bioink. So if you repeat this process again and again, right, you can imagine eventually you can create a 3-dimensional structure with each layer having a different pattern that you wanted, right? So here shows an example of how that process is, right? So basically you can infuse the bioink into this tank, and then you can change a pattern in a layer by layer manner. And then eventually, if you bring the stage up, you're going to create a little three-dimensional structure of complex architecture within this little volume, right? So, and for example, you can then create more complex patterns of vessels and stuff in there. So then one of the issues, as you can imagine here, is that you only have one single tank, meaning that you can only print a single material, right, or a single cell type in a single printing process. So to resolve that problem, again, this is, remember, this is talking about multi-material bioprinting, right? So we then developed a system where you can actually incorporate a little chip device to allow automation delivery of the different bioinks to allow multi-material DLP bioprinting, right? So this then allows you to create more complex structures containing multiple materials and multiple cell types. And a few examples, you can then, for example, create these vascularized tumor models with pretty complex vascularization structures in there, having endothelial cells like vascular cells lining these channels, and then the breast cancer cells within the surrounding volume. And you can create also more complex junction features, for example, the muscular tendinous junctions or bone muscle junctions that are having multiple cell types and complex structures within these tissues that you can print. All right, so those are multi-material bioprinting. So another aspect that I mentioned we have been interesting is the candidate tissue bioprinting. So again, starting with vasculature. So we're interested in vascular bioprinting because vessels are very important components in the human body. And again, essentially, you cannot really find a single tissue or organ that does not have an interconnected network of the vessels. So one of the earlier ways to create vascular structure with bioprinting is called sacrificial bioprinting. And you can probably very easily understand that technique by just looking at its name, right? Sacrificial bioprinting, meaning that you can initially deposit or bioprint a little fiber that's going to serve as the sacrificial template later on. And once you have that fiber printed, you can either cast or print another hydrogel or like matrix surrounding that fiber. And what happens is that you can then stabilize this matrix and then selectively remove this initially bioprinted fiber, right? So then you can create hollow perfusable channels within this tissue-like hydrogel matrix, right? So of course, with bioprinting, you can pattern these channels in different shapes, straight, multiple channels, or curvy channels as well, right? So, and as I mentioned, these are perfusable, right? So you can basically perfuse the entire structure of the vasculature that you print. And remember that these are not only structurally relevant because it's a hydrogel or like a jello-like structure, right? So then it's also biocompatible. So then you can also seed relevant cell types on these channel walls to make them biologically functional as well, right? So these are, for example, endothelial cells you find in the lining of the vessels, and basically you can line these printed channels with endothelial cells, and they can grow into confluent layer, just like what you have in the human system, right? So you can also do this for branching channels as well. And if you do a confocal image, you can see these are actually hollow, intact endothelial layers you can create on the channel surfaces. And what's more interesting here is that if you further culture them for a longer time, you can see their secondary sprouting into the surrounding matrix, right, forming this multi-scale vasculature in the bioprinted constructs, tissue constructs. And if you do a diffusion analysis, so basically if you compare, so on the top, this is a printed channel without any cells. You can see quite fast transport or diffusion of this dye molecules into surrounding matrix. But if you compare that with a channel that is endothelialized, right, so having this vascular cells, endothelial cells on the surface, you can see a much slower diffusion of the same molecule. Because these endothelial cells will actually form tight junctions, which is very similar to what they do in the human vasculature, because they can actually form the tight junction in the human vessels and then slower down the diffusion of certain molecules across this barrier, right? So that's a functional analysis. And then if you remember how these tissues are made, so when you cast the surrounding hydrogel after you print, you can then also encapsulate another cell type into the surrounding hydrogel, right? So now you're not only making a vascularized channel, but also you're making a vascularized tissue, because not only have you the vascular channel with endothelial cells in there, but also in the surrounding gel or matrix, you have another cell type, right? So in this case, we actually made a little vascularized liver model for drug testing. And what's interesting here is that if you infuse your drug molecule through this vascular channel, you can appreciate, of course, how the drug is affecting the integrity of this vascular barrier, right? You can see actually it's not intact anymore, it's being bridged. But also we can see how this same drug is going to affect the surrounding liver cells in a time, dose, and more importantly, diffusion-dependent manner, right? So diffusion-dependent manner is very important because in the human system, as you can imagine, if you take a drug, it's usually going to go through your vessel and then be delivered to the different parts of the body. And then once it's in the local tissue, right, so the drug is going to diffuse out from the vessel. And depending on how far away your tissue cells are from the vessel, there's going to be a diffusion-dependent manner toxicity or efficacy of this drug molecule, right? So, and that's something you cannot model with conventional two-dimensional cell cultures because if you infuse the drug or add the drug into the medium, then every single cell is going to expose to the same concentration of the drug at the same time, right? So that's not really physiological. So that can be really modeled only in this kind of three-dimensional biologically and physiologically relevant model, right? So of course, by modulating the matrix prop, you can also modulate the diffusion profiles. I'm going to skip this slide without further details. And those are basically blood vessels, but then you can also, for example, model lymphatic vessels, right? So you can seed lymphatic endothelial cells into the microchannels that you bioprint. And then over time, again, as I mentioned, they can also spread out into surrounding matrix to form this multi-scale lymphatic vascular system. So model this lymphoangiogenesis process, right? So that's also something you can do. And those are healthy vessels you can model. So how about, for example, vascular disorders, right? So it turns out that you can also model those as well. So in this example, specifically, we were modeling thrombosis where like once you have these channels bioprinted and sterilized, right? So you can infuse human whole blood into these channels and then clot them in there, right? So they're actually forming these blood clots within these bioprinted channels. So now you can create a very simple model of human thrombosis. And if you do a cross-sectional analysis, so here is the histology data of our model showing that you have the surrounding matrix, you have the aggregated red blood cells, the clot in the middle of the channel, and you have these endothelial cells lining the interface, right? So if you compare that with a histological image of a real human thrombus, you can see that they're very similar to each other. I mean that our model is actually very representative of what happens in the human system when you have a clot in the vessel. So then a little more interesting thing here we can model is, as we know in the human system, there are different ways of forming this thrombosis and how they can age. So one way is that if you have a intact vessel wall, right, that's going to prevent the interaction between the blood and the surrounding matrix or the surrounding cells, like a parasite fibroblasts. But if you don't have this intact vessel wall, for example, when you have a damage in the vessel wall, then you're going to have direct contact between the surrounding matrix and cells with the blood cells. So that's going to be allowing thrombosis to happen. And what's more, without this barrier, the surrounding cells can actually migrate into the clot that you form and then eventually deposit these matrices like collagens and other things to age the clot. So if you have fresh clot, for example, most likely you can dissolve that away using some agents, for example, tissue plasmin activator. But if you have aging clot with all the kind of dense matrix already produced, then it's becoming non-dissolvable anymore, right? So it turns out that you can also model this complex physiological or pathological process in vitro in your models as well. So for example, if you have an intact vessel wall, you don't really see any migration of the cells into the interior of the vessel where you have the clot. But if you have the endothelial cells, well, if you don't have endothelial cells, but surrounding matrix, then you can appreciate significant migration of surrounding fibroblasts into the clot and making this clot a H clot by depositing these collagen matrices, right? So then they don't really go away anymore, right? So that's basically showing you can model how you can model this complex processes in vitro in this high content models, otherwise not quite possible previously. So, so a brief mention here is, so these are based on the sacrificial bioprinting, and you can appreciate that these are hollow channels, but they usually have larger sizes, right? So we actually more recently developed the shrinking bioprinting method, where you can actually shrink these bioprinted structures to a smaller scale to improve resolution. So I'm not going to go to details of how this is done, but showing some results here, right? So for example, this is a printed vessel that I just mentioned, right? So this is initially at a larger scale. But if you subject that to a shrinking process, it's going to shrink to about half size of the vessel, right? So then it's effectively improving the resolution to about twice this initial resolution that you could directly print. And before and after shrinking, the channels were both perfusable, right? So it doesn't really affect the perfusability of the channels, so that because the channels are still hollow after shrinking, right? So that's one way of resolution enhancement. And what's interesting, more interesting, is that if you start with a smaller channel, right? So these are actually another method, which I'm not going to go into details, but if you start with smaller channel, that's already about 30, 40 microns, micrometers in diameter. And if you shrink that further, you're going to achieve single capillary level microchannels that are perfusable, right? So these are all the way down to about 10 micrometers, which is pretty close to single capillaries that we have in the human system, right? So about 7 micrometers in diameter. And these are all perfusable tiny little channels within the matrix or tissue that you can form. All right. So then, these are basically vessels that we talk about, right? So vascularization and all the things. But what about other things? Actually going back a little bit. So these are vessels that I mentioned you can perfuse them. So basically you can maintain the viability and functions of the surrounding cells by perfusing these vessels to supply oxygen and nutrients. So with that in mind, when we ask, what if you don't have the complex or sophisticated instrumentation for perfusion or those kind of long-term kind of cultures, right? So then you would need a way to oxygenate these tissues from within, right? So one conventional way that people use is based on these chemicals, but then the issues there is that first, they are not providing sustained oxygen release because these are based on chemical reactions. And second, some of the chemical reactions can be toxic to surrounding cells. So then with that, can you actually harness this perfect symbiotic approach that's adopted by nature to then like into our bioprinted structure for oxygenating the tissue from within, right? So as we know, these are different animals we can always see in the nature, and then rely on oxygen produced by these plants, right? So that's one way. But then on the other side, plants are also benefiting from the animals because their waste and all the things can become fertilizers that then further supply the nutrients for these plants to grow, right? So it's a perfect symbiotic approach or system. So not only in nature, but in the human society as well, right? So if you look at thousands of years of agriculture history, people are taking advantage of these crops that they can grow for food and of course for oxygen as well. But then on the other side, waste products by humans and animals, they can be further fed back into the plants or crops to allow their better growth. And if you happen to have a fish tank, as I do at home, you usually have these water plants in there, which are then releasing oxygen for providing those viability for these fish and also other things in the tank. And on the other side, the waste products by these fish or other water animals, they're feeding back into the water plants for them to grow, right? So then with that, can you actually really apply or adapt these symbiotic approaches that you see every day in our lives to bioprinting and better tissue fabrication, right? So then one natural way that we thought was if you can print these plant cells, or in this case, unicellular algae, then they can function as the natural photosynthetic oxygen generator for tissues that you will be able to engineer, right? So it turns out that you can actually print these algae in very nice structures if you optimize your bioinks. So like 4 layers, 6 layers, and all the way to 20 layers. And from microscopy images, you can see this densely packed algae cells within the structures, right? So, and basically showing that you can print high density algae cells to provide oxygen from within the tissues. And if you do a viability analysis for these algae cells, you can see that the algae cells printed, they are pretty alive, right? So they're don't showing any deaths within the bioprinting structure. So meaning that the bioprinting process was actually pretty benign to these algae cells as well. And you can grow this algae for multiple days as well, right? So, and you can appreciate first the increase in the color by this algae because they are actually growing to higher density, but also on the same time, you can appreciate this formation of oxygen bubbles by this algae, right? So because they are actually producing oxygen in a photosynthetic way. So then with that in mind, you're going to be wondering, for example, how can you modulate their oxygen production with light, right? For example, if you expose this algae to uniform light, they're going to grow uniformly, right? But if you expose this algae to light only from top, they only grow more on top. And the same way for oxygen released by this algae. And by turning on and off light, you can then very precisely modulate how much oxygen is being produced by this algae in the culture, right? So that's one way to also tune the like the oxygen delivery by these algae, bioprinted algae structures. And as I mentioned, then we're using these as the internal oxygen sources for mammalian tissue growth. So then we can embed these bioprinted algae structures within, for example, a liver tissue, right? So then you can see this green color is actually the algae that you print. And then on the surrounding of the algae, you can see these blue dots, which are actually liver cells that you embed in the hydrogel and then surrounding these algae structures. And you can also appreciate that even within the structure, within this tissue structure, the algae was still producing oxygen, right? So as you can see, there's a bubble in here. And what's more important is within the structure, if you have the algae, you can see much better oxygen retention within the liver microtissue because you have this algae producing oxygen from within the tissue. But on the other side, if you don't have the algae cells, but only the liver cells, you can see much less oxygen within the tissues, right? So then if you do a comparison, we do show that you have a much higher cell viability when you have the bioprinted algae structure that's releasing oxygen consistently from within the tissue, and in comparison to when you have not this algae structure, but only liver cells, right? So you have more dead cells here because they actually consume oxygen in a much faster way, so you don't have sufficient oxygen supply. And again, if you remember the sacrificial bioprinting, you can also then selectively remove the bioprinted algae patterns after sufficient cell density of liver is achieved, right? So then you can basically selectively remove this algae structure to create hollow channels that are perfusable within the liver structure. And eventually you can endothelialize, basically make these channels like biologically relevant as well, right? So by seeding endothelial cells, vascular cells within these channels. So now you're making this endothelialized or vascularized micro liver tissue that's having a very high density of liver cells within the tissue, which is something that you can typically not easily achieve. All right. So with that in mind, I'm going to move to the next topic. So then those are based on vascularization that I mentioned, but there are many other tubular tissues in the human system that are not vascular in nature, as I mentioned, right? So for example, in this case, we modeled a breast cancer like tissue within these bioprinted hollow channels, right? So if you see breast cancer cells within these microchannels, they can also grow into confluency. And then what's more interesting here is that after they grow confluent, right, so they cover all the areas of the cell of the channel, they can also grow into the surrounding matrix, right? So modern this invasion process of breast cancer in our system, right? And here is a confocal image showing that process. Basically, not only can you see how the cells are going out, invading to surrounding matrix, but also how they form this inward aggregation, which is something that you would naturally see in the human breast cancer kind of scenario. And it turns out that they are also expressing their basement membrane proteins, and also the invading cells had more expression of this NCAT. It's actually a marker of EMT, epithelial mesenchymal transition, meaning that these migrating cells are actually having a more invasive phenotype than the cells that are in the channel, right? So that's basically showing again the biomimetic properties of this model that you can make in vitro in comparison to the in vivo counterparts. And one thing that we did a couple of years ago was we worked with this Axios space company to send some of these breast cancer models that were printed into suborbital flight. So down the future, we can, of course, expand this into more systematic studies, basically allowing us to understand how space conditions, for example, radiation and microgravity are potentially affecting the breast cancer progression and drug treatment efficacies from that sort of experiments that you cannot really understand on the Earth conditions, right? So that's something we have been also trying to pursue as well. Okay, so those that I mentioned so far were based on the sacrificial bioprinting. So then you can imagine when making these endothelialized or vascularized tissues in nature, right? So how about what if you want to make, for example, standalone vessels that you would also see in the human system, right? So then we can use this method called microphysiological bioprinting, which allows us to print this perfusible standalone vascular system in a single step, right? So I'm not going to go into details again in how this is produced, but you can, like the message here is that you can eventually create these hollow microchannels that are also cellularized, that are standalone for modeling vascular structures, other tubular structures you see in the human system. So here are some examples of the experiments. So you can see by tuning the parameters of the bioprinting, you can control the diameter of these hollow vessels you can print standalone, right? So, and then you can also tune the wall thicknesses as well. And use bioprinting, you can, of course, pattern these in different ways, right? Two-dimensional or three-dimensional or whatever way that you want. And here, as mentioned, you can further encapsulate cells within this hollow vessel wall, because again, it's a biocompatible material, right? So eventually it's going to form as standalone perfusible vessels. And it's a hollow structure, right? So because you can imagine a single hollow fiber that's being printed, so the entire structure is actually perfusible, right? So that's very similar to our human like vascular bed with a multi-layer kind of vascular structure. And you can further build in tortuosity into the vessels. For example, you can, of course, if you perfectly match the printing conditions, it's all straight vessels. But if you start to mismatch the conditions, you can start to introduce this tortuosity or the curvatures into the vessels to model some of the curvy structures that you can see in the human system as well. And these curvy vessels are also perfusible, right? So multi-layer, and they're all interconnected. All right, so another variation is that not only can we print these perfusible blood vessels, but there's another system that I also briefly mentioned before, which is based on this lymphatic vessel, right? So while blood vessels are transporting the liquid and blood and all the nutrients, oxygen from one side to the other side, we also have these lymphatic vessels in the human system, which are single and blinded because they're actually draining the liquid from the interstitial flow to allow homeostasis to be established. Right? So by tuning this bioprinting condition, it turns out that you can also create these vessels that are single and blinded to very nicely model the structure of lymphatic vessels. So now if you create a little chip device, you can print a single vessel that's a blood vessel that's perfusible, right? So like blood vessel in the human system. But on the other side, you can actually print a little single and blinded lymphatic vessel to create a microcirculation there that's typically not present in the previous examples, right? So turns out that if you have a single blood vessel that's perfusing the dye molecule versus if you have two vessels, blood vessel and a draining lymphatic vessel, you can see very different transport profiles of the same molecule, because now you have a different microcirculation system built into the micro tissue microenvironment, right? So then you can do cell drug testing as well, right? So we showed that if you create a little tumor microenvironment with a perfusable blood vessel on one side and then a draining lymphatic vessel on the other side, not only can you see differences between 2D and 3D cultures, as I mentioned before, but now you can also start to look at or appreciate the differences between a single channel configuration versus the double channel configuration. Then this gives you a much better mimetic kind of profiles of how the drugs are transported in the human system, because in the human tumors and other tissues, you not only have the perfusable blood vessel, but also the draining lymphatic vessels, right? So that's one way to model this tissue microenvironment in a better way. So maybe a few couple slides. So one thing is that you can also, if you remember how I mentioned these multi-material bioprinting in the very beginning, right? So you can also combine that with this hollow fiber printing. For example, you can coat these fibers longitudinally, meaning that if you print a single hollow fiber, you can coat the different areas to be having different materials, right? So then you can start to pattern them horizontally or vertically depending on different applications. Or you can also pattern these hollow fibers in a natural way. So the rationale for that is, for example, in the human system, the vessels are not uniform. So coming from the arterioles, they are actually double layer structure, having endothelial cells in the middle and then smooth muscle cells on the outside. But coming to the capillaries, it's becoming a single layer structure, only having the endothelial cells, and going back to the venules, it's again becoming a double layer structure, right? So how to really model this transitional structures that you see in the human vessels or other tissues is becoming also important. So it turns out by tuning further the performances of these nozzles, you can now start to switch between single and multiple layers of the walls for these printed standalone hollow fibers, right? So for example, you can start with two materials, and at some point you can turn off the outer layer. So it's becoming a single layer tube that you print. And at some other point, you can then turn on the outer layer again. So it's becoming, again, double layer structures can basically very nicely switch between single layer and double layer or even more layers of structures as you want in a single printing process with this multi-material kind of printing process. Right, so two examples is you can then print more complex double layer or multi-layer structures. For example, ureter having urothelial cells in the middle and smooth muscle cells on the outside, or double layer blood vessels having endothelial cells in the middle and then smooth muscle cells on the outside. And these were all expressing their relative relevant biomarkers pretty nicely as well. Okay, so so then you can, if you remember the shrinking method that I mentioned earlier on, right? So you can also shrink these bioprinted hollow standalone tubes to a smaller scale. For example, if you start with a larger vessel or the tube that you print about 600-700 micrometers, you can shrink those all the way down to about 100 micrometers in diameter, right? So that's already good. But if you start with even a smaller vessel, for example, 300 micrometers, you can then shrink this all the way down to about 30-40 micrometers in inner diameter, which is something you cannot typically direct print previously, right? So then this is going to give you the chance to model some of the smaller tubular tissues that you can find in the human system now with ease, right? So, okay, so I think I don't have much time, so I'm going to maybe skip this last section, but going all the way back to the last slide. So where we showed that so as I mentioned in the beginning, right? So we are also interested in this in situ bioprinting. So again, the rationale behind this is sometimes you have a wound that needs rapid dressing, and the wound is usually not uniform or is heterogeneous, right? So then if you can have a handheld bioprinter that you can directly hold in your hand to print according to the shape or structure of your wound, that's going to be something that's quite desirable for many scenarios, right? So then we designed a little handheld bioprinter. Basically, you can imagine you have conventionally you have this like desktop bioprinter that's huge and bulky. So now you're basically adapting that to a little compact device that you can hold in your hand, and then by pushing these buttons on there, you can directly start to print on any surface or tissue that you want, right? So then you can start to print, for example, on a wound according to its shape and structure in a way that you cannot really do before, right? So this gives you the chance to directly print in situ on the wound area that allows, for example, rapid dressing of the wound and all those kind of applications, right? So with that, I would like to end my talk by thanking all the wonderful collaborators and also the funding agencies and eventually also the wonderful lab members across the years who have done all the work. And I dig up the photos from 2 years ago, and Jack was here back with us in 2018, who actually also contributed a lot to some of the projects that I discussed about today. Thank you.
Thank you very much. That was a fascinating talk. Thank you for joining us. Do we have any questions? Any questions, just unmute yourself and then you can ask away or type in the chat. I think you've probably answered everybody's questions with your talk, I think. But yeah, okay, thank you so much for joining us.
No problem.
Have a good holiday and everything.
Thank you too. Thanks for inviting.
Okay, thank you.
All right, thanks. Bye.
Okay.